All right, everybody, welcome to a special seminar here at Flathead Lake Bio Station. I'd like to introduce Dr. Emma Rosi. She is a senior scientist at the Cary Institute of Ecosystem Studies in New York, which is a place I worked at long ago for over 2 years. She got her bachelor's degree at the University of Michigan and then got a master's degree at Georgia and a PhD at Georgia. And her PhD was with the same advisor that I had, Judy Meyer. So we were in the same labs, but not really at the same time. She started with as a master's student at Georgia, studying invertebrates with Bruce Wallace. And the thing about her then is she was experimenting with hairy clippers, and I wish I had had a set of photos from them. But every day it just got a little shorter as you worked your way down the number line. So she got her PhD working on sort of mix of toxicology, caterpillars, and urban strains, and then went to the University of Notre Dame for a postdoc and worked with Jen Tank and Gary Limberti. Following that was a professor at associate professor too at Loyola University in Chicago, which was great because it's a fun place that I would visit her in Chicago. Right around then, she and I started collaborating on a project where we worked in the Grand Canyon for 3 years. And so, and then after that, we did a big river project around the West and the Midwest. So I think I calculated 1,500 river miles with Emma in the Grand Canyon. And then probably more like 200 river miles for our large river project. So we spent a lot of time together in the field. And, as Jim was asking me, like, well, what are you going to talk about in your introduction? I was like, what's what I'm not going to talk about that matters? And so, yeah, we have mutually assured instruction there. We were like, tell those stories. And so we had but that was a really successful and fun project because we found, because at one point we were writing a proposal and we realized, I know exactly what these guys want. They want what we did our dissertations. We had very similar dissertations, and we just essentially wrote our dissertations for the Grand Canyon. And they were like, "Oh, this is exactly what we were looking for. Here's $1 million." And so, yeah, that was fun. Since then, she moved to the Cary Institute of Ecosystem Studies, which is probably the premier ecological research institute in the world, really. And like I said, I worked there for just a couple of years as a postdoc, and there she has done really important research on everything from catchment biogeochemistry up at Hubbard Brook, toxicology of pharmaceuticals and personal care products, which she'll talk about today. And then a really interesting research project that I got to visit her in Africa on, where they study the effects of hippopotamus and wildebeest on rivers. And so, that was one of the premier trips I've ever taken in my life, was the opportunity to go to Africa and hang out on the Mara River and run from hippos. And she's here visiting this week. Her husband Steve is on a review panel here, and they've been wanting to come out for a very long time. And she's about to become senior scientist emerita at the Cary Institute. So congratulations on that, and I welcome Emma.
Thanks. When you have a friend who you've spent so much time with introducing you, you're like, oh, what's he going to say about me? So Bob and I have had a long friendship, and it's been great working together. So thanks so much for having me and for introduction.
Tell some stories about her behavior at my wedding afterwards.
After I leave. Okay, so I'm going to talk today about rivers on drugs, and I'm going to talk about I'm an ecosystem scientist. So Bob says he's introduced me and said I work on toxicology. I'm actually not, I don't do toxicology. I say I'm an ecosystem ecologist. I study contaminants that are in ecosystems, but I take an ecosystem approach. I learned it from the University of Georgia. And so that's what I'm going to focus on today. And as Bob pointed out, let's see if this works. Oh yeah, went too far ahead. What happened? Here we go. Okay, I have done research on rivers in the Anthropocene for my whole career. And I have worked on, I don't know how to make that go away, but we'll ignore it. So I've worked on all different things. As a research scientist at the Cary Institute, I don't teach, I just do research. So I have the benefit of having a full-time research appointment. And so I do research on all different things, traditional contaminants like mercury, stream restoration, dams, invasive species, things like BT corn, urban streams, nutrient pollution, river and large animals and river chemistry, atmospheric deposition. And today I'm going to talk about novel pollutants, but I've worked on all different things. So I'm really interested in all types of things. As a soon to be emeritus scientist, I'm actually going to pursue my other love in life, which is art. And so these are some of my art that I put into scientific publications. So so keep your eyes open for my art in the future, but that's not what I'm going to talk about today. So I always like to start my seminars with an acknowledgement, because throughout this talk, I'm going to say things like, I may say things like, I did this thing, but really never, it's never an I, it's we. We did these things. And the we, mostly I want to give a shout out to my graduate students and postdocs, former graduate students and postdocs. They've all moved on to bigger and better things, but I want to acknowledge them and my collaborators. So this is everything I'm going to talk about is a group effort, not just work from myself. Okay, let's start with a quote from Rachel Carson. Rachel Carson wrote a book that arguably changed the world, right? So, she wrote Silent Spring, and this is a quote from Silent Spring. The chemicals to which life is asked to make its adjustment are no longer merely the calcium and silica and copper and all the rest of the minerals washed out of the rocks and carried in rivers to the sea. They are the synthetic creations of man's inventive mind, brewed in his laboratory and having no counterpart in nature. This is even more true today than when she wrote those words in 1962. She was prescient. She really was thinking about the world, and we find ourselves even more awash in synthetic chemicals. Of course, I like this quote because it really synthesizes a lot about what I do, thinking about how synthetic chemicals are going into rivers as a river ecologist. So let's start with some information about synthetic chemicals. This is a paper that Emily Bernhardt and I and Mark Gessner wrote. We basically made the case in this paper that these are the traditional recognized agents of global change. So things like nitrogen pollution, biodiversity loss, population growth, CO2, and increase in agricultural land use. Those are the recognized agents of global change as defined by the Millennium Ecosystem Assessment. And we said, hold on a minute, those are important, but so are synthetic chemicals. And so if you look at and we tag this to the time when Rachel Carson's book came out, so Silent Spring, you can see that there is on the same trajectory, exponential increase in pesticides, in the number of approved pharmaceuticals, and in the green are the dollars, because it's very hard to get the mass of how much synthetic chemicals are being produced. So what we use is dollars instead of mass. And what we argued in this paper is that synthetic chemicals are spread throughout the world. They have ecological effects. They're increasing on the same rate as these other compounds. And so we really should think of them as agents of global change. And just how many chemicals are we producing? Like the diversity of chemicals. This is a paper that was published in ES&T in 2020 by Wang et al. And basically they took the CAS numbers, right? They like, well, how many chemicals are there? And it's basically, over 350,000 chemicals or mixtures are registered. This doesn't include the 50,000 confidential compounds, like that we don't even know what they are, or the ambiguously described breakdown products. So when we're talking about, there's a lot of chemicals out there that are synthetic, we're talking about a lot of chemicals, right? And I'm not even going to begin to touch on all these and what they might, the ecological consequences. So I'm just going to focus on one subset of these. Oops, need to be patient with this. Sorry. Let's see if we can go back. Go back one. Okay, so this is a paper by Person et al. In ES&T. You may be familiar with the idea of planetary boundaries. And so this is a paper that they wrote about planetary boundaries and novel entities. And I just want to point out that what they said is that the safe operating space of the planetary boundary of novel entities is exceeded since the annual production and releases are increasing at a pace that outstrips our capacity for both assessing and monitoring and even understanding the effects of these compounds. So we are producing so many compounds, distributing them into the environment so much faster than we could ever possibly understand what they're doing, which I think is a really good way to think about this novel planetary boundary. So I recommend this publication. Okay, let's see. Maybe I can't use the clicker. It's not fast enough for me. Okay, we'll move this way. Okay, so I want to just talk about aquatic ecosystems and how drugs, so the compounds, the subset of those large number of compounds, how are they entering aquatic ecosystems? So people think, okay, well, it comes from us. Yes, it's true, they do. So wastewater treatment plant effluent contain pharmaceuticals and personal care products. Septic releases, municipal landfills. So, you know, that's where we take our trash, right? You put things in your trash, it goes into a landfill. Not all landfills are perfectly sealed, so they leak. And there's been lots of research to show all of these things are sources. Agricultural use from livestock and manure applications. So we're using them quite a bit in our animal husbandry operations. Biosolids land application. So biosolids is another word for sewage sludge. They renamed it to give it a nicer name, but that's what it is. Pharmaceutical manufacturing facilities are the major source of the major way that you have high concentrations of pharmaceuticals. So right now in this country and around the world, pharmaceutical manufacturing facilities are not regulated. They can discharge pharmaceuticals out the bottom of their pipe. So this is the USGS has done some really nice work. And then there's been work done in overseas. And basically, so manufacturing facilities are a major source, and that's where really high concentrations can be found of pharmaceuticals. And what's challenging about that is that a pharmaceutical manufacturing facility, you know, one day they're going to make codeine, and then the next day they make some other drug, and then the next day they make some other drug. They wash down all their equipment, and it ends up in the environment. So it's like an ever-changing mix of compounds. And then combined sewer overflows and leaking infrastructure, and I'll talk about that some more. So now I sometimes give a seminar where I talk about all that stuff, but I'm going to just skip that, and I'm just going to ask that you believe me that pharmaceuticals and personal care products are now a part of river ecosystems. So, and when I talk about this, pharmaceuticals and personal care products, it's a very large group of compounds. It's at least 1,400, because there's 1,467 pharmaceuticals approved in the US, but that doesn't include all of the fragrances, detergents, cosmetics, all those other things that are found also in the environment, sunscreens, insect repellents. So we're talking about a very large number of compounds. And what's really important for you to get your head around is the fact that we group these together as scientists, not because the compounds are in any way similar to each other, but because they come from the similar source. Right, so like an antibiotic is going to behave really differently than an antidepressant in the environment. Effects are going to be totally different, whether or not it degrades, does it sorb to organic matter, all those things that we as aquatic ecologists might think about, they're going to be really different because the compound structure is really different. So that's a really important thing to remember. Okay, so pharmaceuticals basically have been detected in the environment where there are people. So they've been found in the Antarctic Peninsula, believe it or not, because tourists go there. And so tourists, when they go, they bring their pharmaceuticals with them. And so there was a paper that was done that showed that. They're detected in US national parks. There's a really nice paper that was done by Paul Bradley at the USGS, where they took samples all around the US, and they found hundreds of different pharmaceuticals in rivers and streams. And then a paper that came out in 2022. This was like a really interesting COVID project where some people in the UK sent sampling bottles all over the world. They took samples all over the world, and they found, I know you can't read this slide, but they found all these different pharmaceuticals in rivers all around the world. So this is not just an issue in, one place below one wastewater treatment plant. Pharmaceuticals are found all over the place. Okay. So I'm going to tell you a little bit about some of the work that we've done where we're taking an ecosystem, kind of a load watershed approach to pharmaceuticals. So in cities, like in Baltimore, so I worked as part of the Baltimore LTER site on streams in Baltimore. And cities are basically underlain with aging sewage infrastructure. And in Baltimore, the infrastructure went in around the turn of the last century, so 1900, 1908. And basically it's leaking, right? In fact, I know people that work at the DPW, the Department of Public Works in Baltimore, and they don't even necessarily have maps where the sewage infrastructure is. And so it's definitely leaking, it's old, it's aging. Many cities have this problem. So even though in the watershed that we worked at, that's shown here, the pictures from this watershed, all of the wastewater treatment effluent is actually shipped to another watershed. It's a treatment plant in a totally different watershed. But when we go and we sample and the water looks like this, and if I could just take you there, you would smell it and you would say, it doesn't smell like a normal stream because you're smelling sewage. And so the sewage that we're finding in these streams in Baltimore is just leaks in the infrastructure. So when we find pharmaceuticals, it's not due to wastewater treatment plant effluent, it's leaks like this. So this is a manhole cover that it's kind of hard to tell, but that's like, it blew off every time there's a lot of stuff going in there, and that's all toilet paper and nasty stuff in the woods. So that's these study streams have pharmaceuticals from leaking infrastructure. And this is work that was led by my former postdoc, Megan Fork. And basically what we did is we, in the BES, the Baltimore LTER, we've been collecting samples. I think we're now at like 20 some years. We've been collecting samples for nitrogen and phosphorus and chloride and kind of all these different things, normal, traditional, water quality parameters. So in 1 year, we said, okay, we're gonna ask our technicians to grab a sample for pharmaceuticals every week for a year at all of our sites. And we ran those for 97 different drugs. And then we combined, Megan did this really awesome thing where she combined the stream flow data with the concentrations of pharmaceuticals to get export of pharmaceuticals into Baltimore Harbor. And this is a paper that was published in ES&T a couple years ago. And so this is just an example of what we found. So this is the total concentration of all the pharmaceuticals that we detected. And you can see that over time, the concentrations, sometimes they're higher, sometimes they're lower. It changes over time. But because we have both the discharge and the concentration, we can combine those pieces of information together to calculate load. How much pharmaceuticals are actually being exported into Baltimore Harbor. And to my knowledge, no one had ever done this before. And here is a figure just to give you a sense that along this urban-rural gradient, so these are the rural streams, this is the urban streams, you can see increasing concentrations of both the concentrations of drugs and the diversity of compounds. So those are all different types of drugs, like antihistamines, antibiotics, antifungals, antidepressants. And you can see, and this analgesic, this pink bar, that is acetaminophen, Tylenol. So very commonly detected in our samples. The concentrations increase as you went downstream. And then what we did, what Megan did, was really cool work that she did. So we calculated the estimates, how much was actually getting into the harbor. And if you express that in terms of, because this graph is maybe, you don't know how much a kilogram of acetaminophen actually means. If you express it in terms of equivalents of human doses, what we get is, on an annual basis from this stream in Baltimore is 35,000 doses of antidepressants, 30,000 doses of painkillers, and 1,000 doses of antibiotics getting into the harbor. So even though the concentrations are in the nanograms per liter, well, it's a lot of nanograms per liter over a year going into the harbor. And so this is a very different way. People don't traditionally think about it this way, but I think it provides some new insights into just how much are we talking about going into the harbor there. And then the other thing that we did is we routed these pharmaceuticals through the whole system. So basically what Megan did was we said, okay, this is trimethoprim, this is an antibiotic. And let's look at it as it goes through the whole system, what happens to it. And so we figured out, we know in this watershed, there are this many people, on average, people take, how many kilograms, given how many people are there in that watershed, of this antibiotic trimethoprim, we figured out that it's about 23 kilograms of trimethoprim are taken by the people that live in that watershed. Then we route it through the whole, from human use all the way to the harbor. And basically what you can see is that with trimethoprim, very little of it is metabolized. So trimethoprim, we take it, most of it comes out in our sewage, in our waste products. It goes to the wastewater treatment plant. The wastewater treatment plant does not remove hardly any of it. Most of it goes right on through the wastewater treatment plant, and then it gets into the inner harbor. And so it's about, in this case, about 10 kilograms, or about half of the trimethoprim that people take is getting into the harbor. Now, if you contrast that with this analgesic acetaminophen. It turns out that's this one here. So that's trimethoprim, and here's acetaminophen. People take a lot more Tylenol, right? They take a lot more of this this analgesic. So, but what's interesting is that most of that that's being used is actually metabolized in our bodies, right? So remember I told you at the beginning, you can't assume that they all behave the same. We actually remove a lot of Tylenol. Not a lot of it comes out in our waste. And then in fact, the wastewater treatment plant removes almost all of it. But it turns out that the total load to the harbor is the same, because just a little bit of leaking in the infrastructure, when there's that much coming out, leads to the same load going into the harbor. And this is a really different way of thinking about how things move, but I can see that this is a ecosystem ecologist approach to thinking about how the route of these compounds and how they get to the environment. But it shows you these two different, this versus the trimethoprim shows you that if you want to deal with trimethoprim, you got to fix the wastewater treatment plant, right? Because most of it's coming out through the wastewater treatment plant. If you want to fix the problem with this acetaminophen or Tylenol, you got to fix the leaks in the sewage infrastructure. So both of those are required if you're going to deal with these different drugs. Okay, now let's talk about the effects of pharmaceuticals on aquatic ecosystems. And that's really the area that I have specialized in my career. When I focus on this is as an ecosystem ecologist. Like there are a lot of people, environmental chemists go and they measure the concentrations of pharmaceuticals in the environment, and that's great, we can find them. But really what I care about is, does it matter? What do they do? What do these things do to the environment? So I think of it from the perspective of an aquatic ecologist. You know, pharmaceuticals get into the environment, what do they do? And basically I'm going to give you some of these examples, but basically our research has documented a host of ecological effects. I can't talk about them all today, but I will show you some examples. But we have found that they change bacterial composition, they can change antibiotic resistance, they change diatom community composition, pharmaceuticals can alter the rates of primary production, respiration, denitrification, they can change life history and behavior. So I don't have a chance to talk about all those, but I'm going to give you some examples here. And one of the outcomes of all this research is that we wrote this paper, Aaron Richmond, a former PhD student of mine, we wrote this paper that basically argued that pharmaceuticals and personal care products should be viewed as ecologically disrupting compounds. And so, you know, you've probably heard about endocrine disrupting compounds. Pharmaceuticals and personal care products are not endocrine disruptors. They're ecological disruptors. So they change different things like primary production, ecosystem function, those kind of things. But most of the research is actually focused on the toxicity. Very little research is actually focused on ecological effects. And basically, we pose this idea here that the concentration that is needed for ecological disruption is going to be much lower than the concentration that's going to lead to toxicity or a lethal dose. And the way that I describe this in sort of like an offhand way is to say, like, okay, well, how much ibuprofen do you need to take for your headache to go away? Not very much, right? How much ibuprofen do you have to take for your liver to shut down for you to cause mortality? A lot. Right? So these compounds that we take, they're biologically active at low doses. That's why we use them for drugs, right? And so the fact that we focus most of our research on what is the dose that's going to lead to mortality of an organism may be not quite right, right? We need to think about what is the concentration that's going to alter an organism's behavior or alter an organism's, you know, how they interact with their environment or smell or things like that. So we really tried to make this case that we really need to shift our focus away from thinking about the toxicity of these compounds, but rather thinking about how they might disrupt the ecology. Okay, so what I'm going to do, this is the approach that I have used throughout my career. As an ecosystem ecologist, we do everything from single species lab incubations all the way up to large scale downstream wastewater treatment plants. We investigate pharmaceutical effects at all these different scales, and I'm going to give you some examples of these different scales. And what we've argued in this paper that I wrote with Todd Royer a number of years ago is that if we want to understand the effects of contaminants on the environment, we cannot only just do everything in the lab in beakers, we must do it at multiple scales of inquiry. We have to think about things, what happens actually out in the field versus what happens at like a small scale to a single species. So I'm going to give you some examples across this this range of scale of inquiry in this talk. So the first I want to talk to you about is a study that was led by my former student, Rafael Almeida, who is now at the University of Texas as a faculty member. Rafael came to my lab. He was just a visiting student for a year in my lab from Brazil. He came right at the time that there was a Zika outbreak in Brazil, and he was working with me on the effects of hydropower on the Amazon. That's what we were working on. But he said, well, while I'm here, I want to run an experiment. Do you have any ideas? And he said, well, have you ever done anything with mosquito repellents? And I was like, no, I don't know anything about mosquito repellents. Let's do an experiment. So we did an experiment on looking at the effects of mosquito repellent on salamanders, actually. And what Rafael found is that the basically, this is just a graph here. This is salamander larvae and their mortality rates. So it's just what happened here. I'll just explain it. We looked at two different insect repellents, DEET, which you've certainly heard of, and this other one called Natrapel. Natrapel is a derivative of geranium, and it's a different kind of mosquito repellent. And basically, these are the different concentrations that we did of these different compounds. And I'll just cut to the chase here, that basically, when we exposed salamander larvae to Natrapel, what first happened was their tails were deformed, and then it resulted in very rapid mortality. In contrast, they didn't respond to DEET. There was no effect of the DEET on the salamander larvae. And so we sort of talk about, in our paper, we conclude with this figure, which is, and we also did a bunch of experiments to see whether or not mosquito larvae themselves were affected by insect repellents. And the fact is they're not. So we did the same experiment at different concentrations, but with mosquito larvae. So basically what happens is you can envision this kind of vicious cycle where humans use mosquito repellent to keep mosquitoes off, right? So that they don't get an illness, which is an important thing to do. Then that mosquito repellent gets into the environment, and the picaridin containing this Natrapel, picaridin is the name of this compound, it rapidly kills salamanders. But salamanders are the major consumer of mosquitoes, and mosquitoes are not affected at all by picaridin. So like thinking about this from an ecological perspective, we only did some laboratory experiments, but you can envision this kind of scenario in small catchments where we use insect repellent, and it actually is the predator of the insect we're trying to repel is highly affected by it. So that's just some feedbacks that could potentially exist. Another thing that we've done at the Cary is I have a series of artificial streams. I have 20 artificial streams. They look like that. They're 4 meters in total length. It's a really great facility. We can do longer duration experiments. We can look at ecological complexity. We can add bacteria, algae, and vertebrates, and we can measure ecosystem functions. And so we've done a whole bunch of experiments that I alluded to earlier, the effects, looking at the effects of pharmaceuticals on denitrification or primary production or respiration. We've done a lot of experiments on that, but I don't have time to talk about them, but it's a really useful technique. I'll just show you some examples. My student, Erin Richmond, who I talked about earlier, she was really interested in how antidepressants might affect aquatic insects and aquatic ecosystems. And, a lot of this work that we do actually, there's nothing to even base it on. There's no research, no information about what an antidepressant might do to an aquatic insect. So we sometimes are going blind here. But what we did was she added fluoxetine, which is the active ingredient in Prozac, and citalopram, which I can't remember the trade name for that one. But basically, when we looked at, and then she put nets over the streams, we had a whole bunch of aquatic insects. We put nets on there. We looked at the emergence of the aquatic insects. And what we found is that when we exposed them to low concentrations, and we redid this experiment at even lower, so 20 nanograms per liter of Prozac and citalopram will result in aquatic insects emerging faster. Why? We don't really know, but it seems to be disrupting the life history of these organisms. The one thing that we do know is that aquatic insects will use serotonin as an indicator. It's a life history cue. So if you expose them to a selective serotonin reuptake inhibitor, an SSRI, which is what Prozac is, it seems to disrupt their life history. And so that was these couple papers. Work that was done by AJ Reisinger, who was a postdoc at the time, and Lindsay Reisinger. They led this really cool experiment where we looked at, this is a crayfish here. So Lindsay is an expert on crayfish. And so we did looked at the effects of an antidepressant on crayfish behavior. And so here we did this really neat experiment where we exposed them to antidepressants and look to see how they responded. And you've made it in science when your research becomes a cartoon in the newspaper. So AJ's like, look at this. It's about our research. So basically we exposed crayfish to antidepressants for 2 weeks. And the ones that we exposed to the antidepressants, we had this really cool technique where we put them in this racetrack thing and we could see how they behaved. And they became more bold and more interested in food and less interested in other crayfish, because we did this. I could explain it, but we had this really neat thing. So, but then we did find that crayfish behavior had significant effects on stream metabolism, but the metabolism itself was not affected by the exposure. And so we were hoping to see whether or not it affected the stream metabolism, but it affected the crayfish more. And so that's what this study was about. Okay. Now moving on to so we've done these lab experiments where we take salamanders and expose them to things in jars, and then we can go up in scale, we can do artificial stream experiments. But then the other thing is, because of the fact that so many pharmaceuticals are being used in our everyday lives, we can actually take advantage of what we call natural releases, right? And so when we think about natural urban releases, natural releases of these compounds, we can use, like, for example, urban streams as a gradient to study pharmaceuticals. And this is, again, work that was led by my former student. She now works at the EPA in Australia. We went to Australia. We got funding from the Australian government to do this research, and we sampled food webs below wastewater treatment plants along an urban-rural gradient. And this is a paper that we had published. We screened these aquatic insects for 95 different pharmaceuticals. And then we also measured both in-stream invertebrates and spiders. And then we modeled exposure to predators. And I'll explain that in a minute. And this was published a couple years ago. Okay, took a bunch of bugs, sent them. First, they came to the Cary Institute. They came from Australia. We brought them to the Cary Institute. We weighed them, we ground them up. Then we sent them to Sweden, where my colleague Jörger Fick is the one who analyzed all these samples. And you cannot read this because there were over 68 different pharmaceuticals, 69 drugs detected in the aquatic insects. That is a lot. We only screened for like 100 and whatever I said, 97 or 102. We found 69 different drugs, some of which, the number that we did, the 102, some of which aren't sold in Australia. But so if you think about the fact, remember I told you early, 1,400 pharmaceuticals, at least in the US, are out there. We can't screen. You only find what you look for when you're doing this. And so we found 69 different drugs, all different types of compounds found in the aquatic insects. So do they accumulate in aquatic insects? Yes, they do. And the concentrations are very high. The concentrations in the aquatic insects are 3 orders of magnitude higher than the concentration of drugs in the water. So if you take a water sample, it's in the nanograms per liter concentration. It's 3 orders of magnitude higher in the aquatic insects. We also found pharmaceuticals in the riparian spiders. So we went and Aaron collected spiders along the riparian zone that the only thing that they eat are emerging aquatic insects. So we collected spiders, and they also have between 38 and 62 different drugs in their tissues. And in fact, the concentrations are similar or higher than what you found in the aquatic insects. We didn't have enough data to estimate biomagnification in spiders, but we definitely know that they are in there. This has implications. So spiders are relatively easy to catch. You have to go out at night with a headlamp and look for them, but they really are an indicator species for us, because if they're in spiders, they're coming out of emerging aquatic insects. And what else eats emerging aquatic insects? Birds, bats, lizards. A lot of things rely on these. And what our data show is, and this was for the first time anyone has shown this, that not only are pharmaceuticals in the aquatic insects, they're moving up into the food web. And just people hadn't measured it before. And so now now lots more people are doing it, which is very exciting to see that people are measuring this. Okay, now let's think about what might the implications be if you're a consumer of aquatic insects. The other thing that we have that's really cool, because we work in Australia, is this egg-laying monotreme that you probably haven't thought about since you were in like first grade, which is the platypus, the duck-billed platypus, right? They live in streams in Australia. That is their habitat. They are a stream organism. Their duckbill is for scraping bugs off of rocks. They have these little cheek pouches, and they store the bugs into their cheek pouches, and that's all they eat. And it's like, I've never laid eyes on a duck-billed platypus, but we can write a model that thinks about them. And then there's also brown trout, which have been introduced, which are less exciting. But so what we did, and don't really have the time to explain it all, but basically what we did is we used bioenergetics approaches to figure out, okay, you're a platypus, and you live in this stream in Australia. How many bugs do you eat every day, right? That was already known. That's a literature value. How much bugs is a duck-billed platypus eat in a day? How many insects do they eat in a day? What's the concentration of pharmaceuticals in those aquatic insects, which we measured, right? And so with a little bit of math, we can figure out how much of a human daily dose might that platypus get every day just from eating the aquatic insects in the stream. And then we did the same thing for the trout. And that's what I'm going to show you now. So this is the percent of a human daily dose. So we express it as a percent of a human daily dose because I don't know how much antidepressants a platypus should have. We scale it, of course, right? A human daily dose is based on our body weight, and we scale it to the platypus weight. And what you can see, for example, is that platypus get nearly 60% of a human daily dose of antidepressants just from eating the bugs in the stream. Incredible, in my opinion. The fish, less so, but so antidepressants, because they were very commonly found, and the concentrations were high. We do have to use some caution with these estimates. We do not know if all the drugs that are in the aquatic insects are actually bioavailable to the platypus. I don't really know that. I know that they must be somewhat bioavailable because the spiders have them in their tissues, right? But we don't know. That's something we don't know. But I do want to point out that, for example, in the US, 10% of Americans have taken 5 or more pharmaceuticals in the last 30 days, right? So like that's on average, people take, a lot of people take not very many pharmaceuticals, but a number of people take more than that, right? So if on average, and the fish and the platypus and the spiders are, when we looked for 102, we found 69 compounds. So I always say to people, when you go to the doctor, what is the first thing they ask you? You fill out the forms. The first thing they want to know is, what drugs are you on? What pharmaceuticals are you already taking, right? Because we know that in people, drugs result in interactions, right? We also hear it on those ads on TV, right? Don't take this if you're taking X, Y, and Z. Well, these aquatic organisms, they have no choice. They're taking whatever is coming out of the wastewater treatment plants. And as I said, we found 69 different compounds. So these duck-billed platypus, they have 60% of a human dose, but how does that interact with other drugs, right? How do those interact with each other? We don't know. We don't know what the consequences are for these organisms, but we better start thinking about it. Okay, now I want to talk a little bit about how common might this be. So that was we measured, I think, 10 streams in Australia. And now we've had the opportunity now with my colleagues at the USGS, Travis Schmidt and Paul Bradley, and my colleague Jirka Fick. So Travis Schmidt, who was at the USGS, was leading the NAQUA sampling. So that's the national, I'm not going to get the acronym right, but they went out and sampled for this study that I showed you earlier, a bunch of streams in the Northeast. And Travis reached out to me and said, I'm going to go to these streams. Maybe I could, this was at the same time that Aaron's paper came out about drugs in the aquatic insects. And he said, how about if I collect a bunch of stream insects at all these sites and we run them for pharmaceuticals? And so we did. So we got tissues from 27 different taxonomic groups, from 94 streams across the US. We ran it for 102 compounds. We haven't published this yet. This is Travis and me, and this is in prep, where hopefully this will come out soon. So how common are drugs? We detected 72 drugs out of our screening of 102. A number of the drugs, because they're run in Sweden, are not actually sold in the US out of that 102, but we detected 72 drugs. The most common drugs detected were a blood pressure medicine was found in 53% of the insect tissue samples. An antifungal was found in 60%. Bupropion was found in 62% of the samples. And what's also, so this is the number of compounds detected in aquatic insect tissues and the maximum concentrations that were detected of all different types of drugs. And you can see they're kind of all over the place, especially the psycholeptics are very high, antidepressants are high, the concentrations and the number of compounds detected. We, at the same time that Travis went and collected those aquatic insects, someone took a sample and measured the concentrations of drugs. So Paul Bradley's work on the concentrations in the water, there's no relationship. We found no relationship between what's in the water and what's in the bugs. And that's not that surprising, because I'm sure the bugs are integrating over time what they're being exposed to. So you wouldn't expect it to match perfectly, but we did look at that in the data. The other thing is, we looked at all different types of aquatic insects. So this is a bunch of different groups, including spiders. And we did focus our effort on hydrocyclid caddisflies. We said, well, wherever you go, always collect hydrocyclid caddisflies, because those are really common and they're found in a lot of streams. So that's what that top one is, of mainly hydrocyclids. And you can see that the number of compounds detected and the maximum concentrations are quite high, but we kind of oversampled those. We did that on purpose. They may have a greater number of detects because they're very tolerant of pollution. So they're found kind of anywhere you can find caddisflies. So that would make sense. Similar to our work in Australia, we found a bunch of different compounds in a bunch of different aquatic insects and in riparian spiders. So that, and the concentrations are in line with what we found in Australia. So what we found in our small study in Australia seems to be the case, at least around the northeast of the United States. So we're really excited about these data because it shows that it's a pretty common issue. I didn't explain to you that last compound, bupropion, that I told you was really commonly detected. Probably no one has any idea what bupropion is unless you're trying to quit smoking. So if you're trying to quit smoking, bupropion is a really good smoking cessation drug. What's interesting, I told you that there was no relationship between the concentrations in the water and the concentrations in the bugs. But what Paul Bradley found when he looked and he did screening for the water, it turns out that one of the most commonly detected drugs in the water is nicotine. And then one of the most commonly detected drugs in the tissues of the bugs is the nicotine cessation drug. I find that to be sort of, I don't know, interesting, strange. But yeah, so yeah, hopefully we'll publish this very soon. And then I want to tell you one last thing about drugs in wastewater. You can't see it, but that's, it may affect human health. So here is a little illustration that I did for this paper that we just published this year on the question that we pose, and the title is, are animal disease reservoirs at risk of human antiviral exposure? So here's the idea. We use antivirals to treat emerging viruses like COVID-19, right? We use antivirals. We don't have very many antivirals, but we use them. That's like our first line of defense before we make before we can make targeted vaccines. The antivirals get used by the human population. They go through the wastewater treatment plant. They end up in streams. They end up in aquatic insects. Aquatic insects are a major food resource for bats. Bats are the things that harbor beta coronaviruses, right? So bats are, where these pathogens may result from. So this is work that I did with my colleague Jirka Fick and Barbara Hahn, she is a disease ecologist at the Cary Institute. And so basically, we know that antivirals are used to treat pandemics. And we wanted to pose the question, is it possible that bats might be exposed to antivirals? They do harbor, they harbor them. So the way that we kind of asked or looked at this problem, thought about it, is first we took that data that I showed you earlier from Wilkinson et al., where they had measured pharmaceuticals all around the world. We overlaid the pharmaceutical concentrations with the species richness of bats. And so it turns out bats are definitely present. Bats that eat insects are definitely overlap with pharmaceutical concentrations. That's like, yes, there is an overlap. It is possible that bats that eat aquatic insects live in the same places where pharmaceutical compounds have been detected. That's definitely the case. So there's an overlap. The thing is that depending on the bioaccumulation factor, so this is, do antivirals, are they found in high enough concentrations in the aquatic insects to result in bats getting exposed to antivirals? That is a piece of information we do not know. We didn't measure antivirals in our studies. We haven't measured antivirals, so we don't know the concentration of antivirals in the water. Is that in the insect tissues the same? 1,000 times greater? We don't know. But what we did is we basically said, if depending on the bioaccumulation factor, so we did a couple different models. If the bioaccumulation factor is 1, then the likelihood of bats basically, we did the same bioenergetics model approach where we say bats eat this many bugs every day. This is how big a bat is. How much would they get in their dose? And you can read the paper if you want more information. But basically, if the bioaccumulation factor is low, the bats are going to get like a very small fraction of a human dose of antivirals. If the bioaccumulation factor is more like 1,000, which is what we've seen for other drugs that I remember I showed you, the concentrations are 1,000 times greater than the water, then the bats would consume about 200% of a human dose of antivirals if they're moving up through the food web. The sweet spot, but bad sweet spot would be somewhere where the bats are being exposed to antivirals that are lower than the concentration needed to actually kill the viral infection, right? So you know this from yourself, right? So, you're supposed to finish your course of antibiotics. You got to finish your course of antivirals, right? You don't want to take a small amount of one of those because then you end up with resistant infections in your body. And so we're very concerned that basically this could result in the spillover of antiviral, a spillover of a antiviral resistant pathogen to humans, right? So we don't know this paper is sort of I joke, there's like a data-free paper, idea rich. We don't have any of the information. We need someone to go out and actually measure these. But what we say is that it would be very prudent to limit our release of antivirals to the environment. Because if antivirals get into the environment and get into bats, and bats harbor diseases that spill over into humans, we really should try and make sure that that doesn't happen. So you can read that paper if you're interested. And especially in the past, when we've had major viral infections, as I told you, remember, manufacturing facilities are a major source of, this has been the case with antivirals, concentrations coming out of manufacturing facilities can be very high. Okay, so rivers have a mix of drug cocktails. As I told you, 1,400 pharmaceuticals in the US. Plus, there's all those other stressors that you probably all know about: nitrogen pollution, salt pollution, climate change, invasive species. All those things could likely interact to have cause effects of these pharmaceuticals on aquatic ecosystems. I'd like to end my talk with, it's to be continued. The effects of pharmaceuticals on the environment, we definitely know that they can have effects at the concentrations. We know that they're present in the environment. Now we know that they're present in food webs. We know that they do things, but there's so much more. I feel like in my career, I've merely scratched the surface of this issue because there's so many different types of compounds, so many different interactions that could happen in the environment. Now you might be asking yourself, aren't these pharmaceuticals and personal care products regulated? Please, we have the Clean Water Act. Are they regulated? No, they are not. I'm sorry to say. The EPA regulates the release of sewage, but not pharmaceuticals. They are not on the list of priority contaminants in the US. So they're not actually listed as aquatic contaminants. So they're not, you don't need a NPDES permit if you're a manufacturing facility. You don't need a permit for pharmaceuticals. The FDA, the Food and Drug Administration, is the one who regulates the safety of drugs, but they don't regulate the environmental release of drugs. So they just think about, are they safe for us to use? The EPA is the one who would regulate that, and they're not considered pollutants. So the effects of the pharmaceuticals on the environment are not well understood. I mean, they are better understood now that we've done a bunch of research in others, but they're certainly not well regulated. But what can you do? What you can do is you can reduce your use of especially personal care products, not of pharmaceuticals, right? Obviously, finish the course of antibiotics. If you need medicine, you take your medicine. But, sometimes, maybe you don't need whatever, that extra dose of DEET or, different things. So reduce your use, especially when it comes to painkillers and things like that. Definitely properly dispose of unwanted medication. And this has happened to me a number of times where I give a talk like this, and it turns out that this Saturday is the National Take Back Program. So you can, and here are places in Montana. So I looked it up, right? 50 miles away from here, you can take your unwanted medications to a proper take back program, and they will dispose of them. They will incinerate them. So DEA every year has an annual take back program. and something the DEA like reports how many they have taken back, and it's many, many millions of pounds that they have collected of pharmaceuticals. So please dispose of them if you have them. And then, of course, urge lawmakers to reduce and upgrade infrastructure. I will get to your question in just a moment. And then that is it. I'm done speaking, and I'm happy to take any questions. So thank you so much for your attention. Yeah, go ahead.
I was just gonna add that we have in Flathead County, we have 9 drop-off locations.
Excellent. Excellent.
So there are 3 at the Whitefish Police Department, Columbia Falls Police Department, Flathead County Sheriff's Office. If you go to wastenotproject.org, our website, it lists all the drop-off sites, and it's really important that that's a simple thing we can do.
Yeah, very simple, easy thing. Don't put them down the drain, please. So, yeah, Bob, go ahead.
So platypus and bats are smaller than people in doses scale allometrically. So do you guys consider that in these?
So we scaled by weight because that's how we scaled it, right? We scaled it by weight. So we like said, well, the weight of a bat is a lot smaller than us, so we scaled it.
Okay, but the dose would scale, so like when
Yes.
when they feed a mouse drugs before they give it to us, they're not gonna give us the same amount of drugs they gave a mouse.
Right. So we
Of
scaled it
scale
that way.
for
We scaled,
weight.
yes, we scaled it for the weight. When we talk about the daily dose, we scale it by weight.
Yeah, but what about the allometry, the fact that if you,
Yeah, I would say that I don't think that we scaled it that way. You
Yeah.
could. There's not, I guess you could do it that way. That's not how we did it. We just scaled it down by weight and said, but we recognize the other thing that's important to remember is I have no idea what the effects are. I mean, a platypus also is really different from people. So
sure.
there's also that,
Yeah.
right? So, but we scaled it.
But the mouse literature ought to have it, because all drugs get fed to mice before they get to
Yeah,
people.
yeah. That's a good point. I don't think we did that,
Yeah.
but good points. Sorry, I didn't repeat his question. I will try and remember that for the next question. Yeah, go ahead. Oh,
You also look at recreational drugs.
yes. So the question was, do we look at recreational drugs? Yes. So we have a couple of papers. So we did a study. I didn't have time to talk about it. Again, you could look at my website. I've done a lot more than just I could focus on today. We have looked at amphetamines. We have a paper published in ES&T, Lee et al., where we looked at amphetamine use and not amphetamine use. We've measured the concentrations of amphetamines in Baltimore streams. We detect amphetamines. We ran an artificial stream experiment at the concentration we detected in the environment to look and see what the effects were. Amphetamines cause aquatic insects to emerge sooner. It changes the algal community structure. It changes the microbial community structure. All those things. So we did pyrosequencing of the microbial community. So yes, amphetamines does change it. We have done that. So that was the only illicit drug that we have run experiments on. We also have, but we've also looked for illicit drugs. So my colleagues and I have looked for them in streams, and you can definitely in aquatic ecosystems. And we have found amphetamines. We found basically morphine, which is what happens when you take heroin, your body turns it to morphine. So what comes out is morphine. So we've detected morphine, we've detected cocaine. So we have MDMA, we've detected those in streams in Baltimore and other places where people have looked. So we definitely find them in the environment, and we have shown that they have effects.
I assume also the waste chemicals of those persons.
Yes. Yeah. So say again.
I know in the Netherlands they track those waste chemicals to processing places.
Yes.
They follow those.
Oh, to follow and to find, but yes. So we have not done that. That's called like, like you can do surveillance to try and figure out where things are coming from, like potential sources of pharmaceuticals. We haven't done that. I don't do that kind of work, but we have found them in the environment for sure. Yeah. Matt, did you have a question?
I did. So, great. This is fantastic.
Thanks.
How are the bugs getting these drugs? So I assume from their food, but how does the food get the drugs? Things they eat,
Actually,
how bacteria or algae.
So they're getting them from the water. So I haven't done this work. My colleague Jirka in Sweden and someone else, sorry, forgot his name this moment. They've worked on basically bugs will equilibrate with the water very quickly. So they're getting it from the water. The aquatic insects are getting it. It's passing into their bodies very quickly. Same with algae. It's like passing into their bodies. Now, not every drug is gonna behave the same. So some of the drugs we're probably never gonna find in aquatic tissue. And other drugs we'll find in, in algal cells and never find in aquatic insects. So it's gonna depend, but they're definitely getting it through the water, not necessarily only through the food, because they will equilibrate pretty quickly with the water. If you put a bug in water with drugs, they will end up with those drugs in their tissues very quickly. Do I understand all those processes and mechanisms? I do not at all. Do we need people to do research on it? Yes. So that's not really what I do, but there's much more research needs to be done on that area. Yeah, it's a good question. Yeah.
Did you do any research, or do you have any plans to do research on any of those drug compounds maybe being found in the tap water in different cities?
Yeah, that's a great question. That's a great question. So I don't do research on tap water, but there have been a bunch of surveys where people have looked at tap water or looked at drinking water facilities in particular. And traditionally what we find, at least in the US, is that our tap water tends to be extremely low in pharmaceuticals and personal care products, in the nanogram per liter concentration, very low. Why is that? Because we don't take our drinking water downstream of sewage effluent. We pretty much know, let's take it upstream before we discharge our waste. So most of the concentrations are really low in the water. And I would argue that actually what's in our tap water, we know that 500 milligrams of ibuprofen is fine. So if there's like a nanogram of my ibuprofen in a liter for water, that's not really going to be a problem in terms of our health. Now, that is in the US, that's work that's done in the US and in Europe, they've done that. But I do want to point out that most of the pharmaceuticals that we purchase in this country are produced elsewhere in the world. And that is not always the case in other parts of the world. Like the concentrations are not always low in potential areas where people take drinking water. So I'll just give you an example. Many of our drugs are produced in Hyderabad in India, because that's a city where a lot of know-how for pharmaceutical manufacturing has occurred in this one city. And my colleagues from Sweden have taken samples in the river below a whole bunch of, there's like 70 pharmaceutical manufacturing facilities. And that river, like 1 liter of water will provide a human dose. The concentrations are just really, really high there. So that, it's not only what we find in the US, other parts of the world. And it's interesting because, Michael Pollan taught us all, the omnivore's dilemma, think where your food comes from. Unfortunately, there's not that same ethos for where do your drugs come from. And in fact, if you go to the pharmacy and you pick up a bottle of ibuprofen, you have no way of knowing, was that produced in upstate New York or India or where? What does it come from? Like we don't know that. And yet the regulations and how people discharge and how things are manufactured are really different around the world. And so again, manufacturing facilities are a major source of pharmaceuticals to the environment. And that's especially where you find the concentrations really, really high. So I answer your question and giving you lots more information. So, yeah. Thanks, Nanette. Yeah.
Along the lines of this gentleman's question, for thinking about cities that recycle water, like Denver,
Yeah.
7 times, right?
Right.
How about in situations like that? I don't know the process they use to recycle that water, and I presume it's fairly clean, but maybe some of these treatments don't capture some of these drugs where some are.
Yeah, that's a great question. So the question was about what about cities that recycle the water? I don't really know. Probably there's research out there. I haven't kept up on it because I don't focus on the effects on drinking water. But often if cities use reverse osmosis, it may remove the drugs just fine. Again, I like to point out that I think drugs in our drinking water is probably not where we should focus our efforts and concern, because again, the concentrations that we can handle are way higher than what's in the water. But if you're an aquatic organism and you live in the environment, you have no control over that. We're releasing this kind of, higher concentrations, and they're being exposed to a whole bunch of different things at concentrations that we don't know what the effects might be. So yeah. I'll take a question in the back.
I was wondering about when you said changes in algal communities, are you seeing like just decreasing populations or changing populations? Or...
Yeah, thanks. The question was about, when I was speaking about changes in algal communities. We've done a couple of different things. We've both looked at the composition of algae. I was fortunate to have Sylvia Lee, who is an expert in diatom. She teaches the class on diatom, so she was a postdoc in my lab. So she identified a bunch of all the diatom species, and she showed the exposure to amphetamine changed the composition of the community. It also changed the primary production. So we both looked at function and structure, and we found that both of those were suppressed. And then we have a couple other different studies where we show that different drugs suppress the rates of primary production. So we both have looked at structure and function of the communities. And we found that some of the drugs can disrupt both these. Other drugs, some of the drugs we've studied didn't do anything to primary production, rates of primary production. So again, it's really important not to generalize, like drugs do this. It's like, well, this drug did this thing and it didn't do this other thing, right? So we try and be real careful about, the pharmaceuticals that we study. If you're talking about ibuprofen versus diphenhydramine, which is the active ingredient in Benadryl, versus amphetamine, those are completely different compounds. They're going to affect completely different things. And so we study the effects on different endpoints, and sometimes they have effects on one thing and not another. And I don't really think you can generalize, except for that I can say that these compounds are biologically active. They have effects at very low concentrations on a whole bunch of different endpoints. That I can say. Yeah. Laura, you had a question?
To what extent in these recent projects are you targeting streams that are like low volume and potentially high source concentrations where you're expecting to find things versus including like maybe rivers that are less altered? And then I have another question, which is like, how close are you to feeling like you could provide recommendations for different, communities or groups or whatever that are looking to monitor this in terms of passive water versus invert versus maybe some kind of passive sampler as like the appropriate way to go about sampling and monitoring?
That's a great question. So for people on Zoom, the question was, did we select for sites that we thought the concentrations were going to be high, or were we sampling a bunch of different things? So first I'll answer that one. The NAQUA study, so in both our study in Australia, we went along an urban rural gradient. So we went to a national park, a stream with a national park, all the way to streams below wastewater treatment plants. In fact, we even saw the drugs in the aquatic insect tissues in a national park because there were people in the watershed. So it's hard to find places where there's no people at all. But we looked at the gradient, but you can definitely see the concentrations are super low in the national park aquatic insects versus below the wastewater treatment plant. In the recent paper that we're doing, that Travis is working on with me, that's just they just went to 94 different sites, all different types of sites. We were not targeting places below wastewater treatment plants. So that's one thing that I think is really powerful about that study, because we went to a whole bunch of different places and looked, and we found very commonly we find these drugs. And again, because in the Northeast part of the United States, you're hard pressed to find a watershed that doesn't have a septic or some kind of, human footprint on it. So that's one answer. It would be great to do national surveillance all over the place and even go to places where there aren't people. You probably won't find these compounds. In terms of how far are we to recommending to people about what to do? So that was the second question. Like, are we at a point where we can make recommendations about either monitoring or control? My recommendation is typically, so in order to measure these compounds, it's very expensive. It is very, very expensive. It is challenging analytical chemistry to measure these compounds. I say it like, oh, we measured 97 compounds as if it was easy. It is not. It is very, very hard. So my colleague in Sweden does a really good job. It takes a long time. The QA/QC is very hard. And so I don't necessarily think that people should go out and measure pharmaceuticals. I actually think your efforts are best if you're like a municipal trying to make decisions about how to allocate resources. I would say the best thing to do is reduce your wastewater effluent. First, fix your pipes, leaking infrastructure, make sure the pipes aren't leaking, and then upgrade your wastewater treatment plants. So wastewater treatment plants, and if you talk to wastewater treatment plant engineers or people that manage the infrastructure, they'll say, we're like one of the last places to get funding. It's typically local government, municipal funding. That's how we fund our wastewater infrastructure in this country. And they're often kind of left, they always want to upgrade their wastewater treatment plants and not necessarily the resources to go to them. So I would say rather than investing resources in, going out and trying to measure these things, you're better off fixing the leaks in the infrastructure, because then you're gonna have benefits for nutrient pollution, pharmaceuticals, anything coming down the pike. The other thing is, they're not regulated compounds. There's so many of them. There's 1,400. If we just spent we could spend forever, one after another, trying to regulate them. That is not a good solution. I think the best solution is to upgrade our wastewater treatment plants to be the best possible, to remove everything, so that then the new compounds that get discovered 3 years from now also get removed in the process, right? So we're not always playing catch up. This is a problem that we could in a very straightforward way, get in front of if we treat our waste better, in my opinion. So that's what I recommend to people, is invest in your infrastructure. Wastewater treatment plants are doing a tremendous service. So I'm over time. I'm sorry about that, Bob.
Last one, Richard, and then Emma's gonna be here for a while, so feel free to hang out now, but yeah.
So you measured exposure to wildlife on several occasions. Have many people measured concentrations in wildlife?
That's a great question. So the question was, have people measured concentrations in wildlife? Like, no one's ever measured like concentrations in platypus. We have, my colleague has done, well, maybe people are trying to do it now that we published that paper. I hope someone does it. No one has measured, we have measured some concentrations in fish. So my colleague in Sweden has measured concentrations in fish. It's very interesting. These compounds are unlikely to biomagnify in a traditional way, because fish have livers, and we know that our liver, like, you know how I told you some of these drugs are metabolized, probably the same processes that exist in our bodies exist in fish, but that doesn't mean it's not going to have some effect on their behavior. So we know that fish can, when they're exposed to different drugs, will change their behavior. They might not accumulate it in their tissues like mercury, but if it alters their ecology, that's something to consider as well. So I'll end with that.
All right, thank you very much, Emma, for a fantastic. Please stay and hang around for snacks and drinks.